Today we're going to talk about Salmonella. So Salmonella you might know as those bacteria that give you diarrhea if you eat undercooked chicken or from the raw cookie dough. But also those bacteria that cause typhoid fever or enteric fever. And so we'll ask, can this give you this one? So we will go into the different types of Salmonella there are, then in those very different manifestations of enteritis versus typhoid fever, and we'll really try to understand why different Salmonella can give you the one and other ones give you the other. To start, here are a couple of motivational questions. I'm considering them optional because we're not going to go into depth really answering them, but you should see that they're helpful for understanding and following this lecture. So question number one is, what are bacterial surface factors or surface markers often used in diagnostics? Question two is, what causes fever? So in very general terms on molecular mechanism. And then where does Salmonella fit into our diagnostic tree? So take a couple of minutes, pause the video, and try to answer those. And yeah, for the first one, we'll this one is obviously relevant for when we distinguish between the different Salmonella, and this one you will also then hopefully find useful when we talk about symptoms of the diseases that we're talking about here. So yeah, just pause the video a little bit. We'll go over these answers only very quickly. I hope that for the first two questions you will see that they are relevant for what we're going to talk about later. The third question is here more for a reminder of the rest of the course and to fit it into the rest of the course. Even so, today we're not going to talk about diagnostics otherwise. So question number one, what are the bacterial surface factors often used in diagnostics? So this is the O antigen, which means LPS, the H antigen, so the flagella, and then often or sometimes there is also a capsule antigen, often denoted then as K antigen. Actually, in the Salmonella, we'll see that the capsule has a different letter, a different combination of letters. What causes fever? So what I want you to think about here is mainly those cytokines that act on the temperature regulatory center in the hypothalamus. These are mainly IL-1, IL-6, and TNF-alpha. Where do they come from? They are secreted by immune cells. Macrophages are important ones here, and they are secreted upon an inflammatory stimulus. For example, lipid A portion of LPS is an important one. Okay, and then let's talk about this diagnostic tree very quickly. Even so, as I said, otherwise in this lecture we're not talking about diagnostics. Okay, so what does our diagnostic tree look like? So Salmonella is in the gram-negative part of our tree, and here within the gram-negatives we have bacilli or rods, which we distinguish in aerobic and anaerobic. The aerobic ones we further distinguish in lactose fermenters, those non-fermenters. Salmonella is within the lactose non-fermenters, and here we further distinguish in oxidase positive ones, and Salmonella is among the oxidase negative ones. So we here further distinguish in those that produce H2S on TSI and triple sugar iron medium or similar medium that allow the production of H2S. So Salmonella we find here among the oxidase negative ones that produce H2S on triple sugar iron medium together with Proteus. Up here at the lactose positive ones we have E. coli, for example. Oxidase positive ones among the lactose negative, here we would have Pseudomonas aeruginosa. Non-H2S producers among the oxidase negative ones, these ones would be Yersinia and Shigella, for example. And here in the anaerobic ones, we would have Bacteroides. Main motivating question was this very naive question. Okay, can I get, typhoid fever or enteric fever from those Salmonella that I consume from, the eggs or the insufficiently cooked chicken? The answer for this is obviously no. They are different Salmonella serovars, and we'll get in a bit to what serovars are. So the actual question, the first main question that we want to ask here is, why are those diseases, so on the one hand side the enteritis from those non-typhoid Salmonella, the ones that you get from the chicken eggs, versus typhoid fever or enteric fever, why are those diseases so different, even though they're caused by bacteria of the same species? But before we go into that, let's define first these different Salmonella serovars. Let's get started, and my hope is that by the end of this class you'll also say that Salmonella are some really fascinating bacteria, that in terms of their mechanisms, they're almost up there, almost as impressive as Staph aureus, I would say. And I also think that studying Salmonella, especially then when we come to the typhoid fever, that this is something that is relatively generally applicable and tells you a lot about medicine and about infections in general. Okay, let's get started. Let's get rid of a first misconception. So Salmonella doesn't have anything to do with salmon. So salmon is not a particularly great source of Salmonella infection. Instead, the name comes from David Elmer Salmon, who is the guy who detected them first, or as it is said, at least his assistant was the one that detected Salmonella first. Okay, so if you've never taken much microbiology, then maybe the first thing you think about when you think about Salmonella is the raw cookie dough. So your mom telling you, don't eat the raw cookie dough because you will get Salmonella infections. And as we will then see later, of course, you don't get the typhoid fever from this raw cookie dough, but you might get enteritis. So as we will talk about later, these are different strains, the typhoid strains and the enteritis strains. So infection sources for those enteritis Salmonella, eggs are an important one. And there is all of this discussion, how should you store your eggs? Should you store them in the fridge? Should you just store them outside somewhere relatively cool and dry? And so in Europe, it's actually, or in many European countries, it's actually the case that in the supermarket you find the eggs not in the fridge, but you find them outside. And this is because people say it's very hard to keep the cooling if you would try to keep a cooling chain intact, that's hard. So there may be a case when you actually get the eggs out of the cooling during transport or something, and then because they're cold, there will be condensation on the eggs. And since these eggs still have their natural protective layer, this would actually be more damaging than if you just transport them at room temperature. This is because in these European countries, the eggs are not washed for this reason, that you want to keep this protective layer on top intact. In countries where the eggs are washed, they would then actually also be transported and sold cold. In Europe, the recommendation is still then once you bought your eggs, once you get them from the supermarket, have them in your house, the recommendation is you put them in the fridge, even so this doesn't seem to be so super important. but avoid the washing the eggs or having condensation on them so that this protective layer is damaged and then keeping them at room temperature. This would be a huge problem. Yeah, so eggs are an important infection source. So in general, coming from chickens, chicken is an important carrier of this Enteritis Salmonella, but also other animals. So there are, for example, these cases when people get infected from their pet reptile, from their pet iguana or something like that. They get a Salmonella infection. In terms of food, the eggs are actually not the most important source. The more important source seems to be chicken itself, so chicken meat. And a possible case would be undercooked chicken. But what seems to be even more prevalent, or the typical kind of thing, is you cut your raw chicken somewhere, and then the chicken goes in the oven, and you bake it or cook it well enough. But on this board where you cut your chicken, you then next cut your salad without having washed the board, and so the salmonella are in the board, they get transferred to the salad, you eat the salad raw, and then this is your source of salmonella infection. In Europe, so this also links a bit to this egg issue, in many European countries the chickens are actually vaccinated against salmonella, so the risk of salmonella infection is relatively low, even though it's not zero. In other countries where this doesn't happen, it's actually not so infrequent that salmonella are found on meat. So the question is then, okay, why don't people not get salmonellosis or this Salmonella enteritis much more frequently? And the answer, of course, is a simple one, that if you use proper hygiene practices in your kitchen and you cook the meat well, then the cooking, of course, kills the salmonella and gets rid of them. There is relatively many sources say there is a relatively high infectious dose for salmonella, even so the sources are quite varying. So what I found is, so the typical range for salmonella is 10 to the 4 to 10 to the 6. Some sources say 10 to the 3. And also, in generally, there seems to be an agreement that the typhoid salmonella, you need a lower infectious dose, but even this is not absolutely clear everywhere. So you can say, if you compare this to something like the E. coli O157H7, then this indeed is a much higher infectious dose. The infectious dose also seems to somehow be linked to several other factors. So, for example, some sources say if you eat or if you consume this salmonella together with very fatty food, then they are protected more and the infectious dose gets lower. As we will see, salmonella, from this enteritis salmonella, well, you get enteritis, so you get diarrhea, but not much more, except in immunocompromised people, for example, they might get a systemic infection. So this is a risk factor then for them, that also, they will have, of course, a lower infectious dose to get the systemic infection. And one thing that's also always cited is gastric acid. So what you find in the classical sources, in the classical microbiology textbook, is that salmonella is rather acid labile, which distinguishes it from Shigella, which is pretty acid stable. This means salmonella should have a high infectious dose. And then what seems important is people taking antacid, some kind of medicine, proton pump inhibitors that reduce the stomach acidity. They can also be infected by a lower infectious dose. I'll answer this question about the different types of salmonella and ask which ones give you enteritis and which ones are the ones responsible for typhoid fever. So broadly, we classify salmonella into two groups. The one is the non-typhoid or enteritis group, and then, of course, on the other hand side, there is the typhoid group. And there are more than 2,500 so-called serovars of salmonella. We'll soon get to what that means. And the two important ones in the typhoid group are Salmonella typhi and Salmonella paratyphi. Then in the non-typhoid group, there are many, but the most important ones, the most prevalent ones, are Salmonella enteritis and Salmonella typhimurium. So the second one may be a bit confusing, that this is actually in the non-typhoid rather than in the typhoid group. Okay, something you may have noticed is that I used capital letters here, which is strange because if you think about, for example, E. coli or Vibrio cholerae, then here the species is always abbreviated with a lowercase letter. Okay, let's get rid of those ones again because, of course, they're not within the Salmonella typhoid group. So the reason for this is that what I'm showing here in the Salmonella, this is not actually a species, but what is indicated by this capital letter now is the so-called serovar or serotype. So the proper way, for example, of saying Salmonella typhi would be Salmonella enterica, and then serovar typhi. So essentially all or almost all of the relevant disease-relevant Salmonella species actually belong, Salmonella serovars actually belong to the species of Salmonella enterica. If we wanted to be even more correct, then we would have to say Salmonella enterica subspecies enterica serovar typhi, but this is something that you often see with the Salmonella enterica and then just typhi or serovar typhi. And what I've shown up here is something that we also typically find where then this capital letter indicates that this is not actually a species, but a serovar or serotype. And now what does this mean, serovar? You can essentially think about it like a bacterial strain, but bacterial strain that is distinguished from other strains based on surface markers, so on serotype, so something that can be detected by antibodies. And the most important ones of the surface markers, this is also by us this motivating question in the beginning, are the O antigen, so O for the German word of Oberfläche, which is, so the O antigen is LPS. Then we have the H antigen for German Hauch, this means the flagellum. And now within the Salmonella, we don't have a K for capsule, but we have actually VI for capsule, so virulence capsule. And this VI antigen is actually only found within species of the, within serovars of the typhoid group. Let's first talk about Salmonella caused enteritis, so the one caused by the Salmonella that you might get from undercooked chicken meat or egg. The symptoms here are essentially diarrhea, so it's a watery, partially slimy and potentially bloody diarrhea. That's it mainly for the symptoms. A systemic infection is possible, however, is only expected in certain groups, such as in immunocompromised or as in young children. Okay, so what's occurring here, what this picture shows is a section of the gut, and up here we would have the lumen, and down here we would have the so-called lamina propria. What is shown here is a so-called Peyer's patch, so a collection of immune cells. And so the Salmonella, they are taken up with, for example, this undercooked chicken meat, and they are here in the lumen of the gut, and then they are taken up through these cells here, which are so-called M cells or microfold cells. So these are cells that are sampling from the gut lumen, sampling antigens, sampling in some cases complete cells, and passing them on through these immune cells in the Peyer's patch, so to these dendritic cells, macrophages. Now, Salmonella are good in surviving inside of macrophages. So using their type 3 secretion system, they can inject proteins, toxins that then stop the fusion of the lysosome with the phagosome. So they can keep surviving in those phagosomes, which are then referred to as Salmonella containing vacuoles. The Salmonella will eventually destroy those phagocytes and be free on the lamina propria side. They can then, from the lamina propria side, enter also the gut lining cells, these enterocytes, lead to destruction of these enterocytes, and they are also recognized by pattern recognition receptors, which are only on the lamina propria side of these cells. It wouldn't make sense if they have these receptors on the lumen side, because obviously they are bacteria, but they have them on the lamina propria side, and this leads then to local inflammation. Local inflammation will lead to an increased permeability of the gut, so meaning liquid is going to flow out here, and also to destruction of these enterocytes, so involvement of granulocytes, of neutrophils that then are going to destroy both bacteria and also the own cells, the cells of the gut wall. And through this destruction, the diarrhea can be not just watery, but in some cases also bloody. Okay, let's now talk about typhoid fever. Typhoid fever is often referred to as a cyclical infection, which maybe is not the best description, but at least you can say that it develops in stages and that it's rather long-lasting. So let's look at what these stages of typhoid fever are. Typically, people refer to 4 stages, which is first the incubation period, then a prodromal stage. So prodromal stage means symptoms are not yet fully developed. So there are symptoms, but they are not fully developed. So typically the fever has not risen as high as it will get during this infection. And often it also means that is rather general symptoms, but not necessarily the very disease-specific symptoms. Then there is the acute stage, or also referred to as the peak stage. And then there is the defervescence stage or convalescence stage. So convalescence, you know, that's recovery. Defervescence stage means a decreasing of fever. And yeah, let's look at this also in terms of the fever curve. So on the x-axis you see the time, which will be shown in weeks, and then on the y-axis the temperature or the fever between an approximately normal body temperature and like high fever around 39 to 40 degrees. And of course, I should say this is not the same for every patient. So both the timings that I'm going to show and the exact temperature that's reached will of course differ from patient to patient, but this is like what I'm going to show is like a typical course of this typhoid fever curve. Let's start with the incubation period. So incubation period means no symptoms and therefore also no fever. So we'll just be at regular body temperature, and then during this prodromal stage we will have an increase up to maybe at a bit high, up to like 39-40 degrees typically. What I didn't show here, often this is a referred to as a stepwise increase. So you may have fever, then the fever will go down again, maybe even to normal body temperature, then rise to typically a higher temperature, and so it will keep going up. Often it's also the case that fever in the evening and then during the day no fever, probably just related to the typical cycling of body temperature as well. So body temperature is highest in the evening, and therefore also the fever is usually seen in the evenings. So incubation period, this was about 1 to 2 weeks. Prodromal stage is around a week. And then we have the acute stage, and this is where the fever stays high. So no more stepwise increase, no more the fever is only in the evening, but now you will have a high fever, as I said, 39, 40 degrees centigrade. Stay high, and this is again approximately 1 to 2 weeks. And then if the patient is lucky, we will have a decrease in fever down to approximately normal body temperature again. And unfortunately, typhoid fever is, of course, a disease where not everybody recovers from, so people might also die here, especially if it remains untreated. But so if there's a recovery, then about 1 week of recovery time. So this is the approximate typical fever curve. Let's look a bit more at the symptoms that we're expecting. And during the incubation period, as I said, incubation period means no symptoms. So this is approximately, the time from the onset of infection, so when the bacteria are actually entering the body, until we see first symptoms. And so symptoms start now with the beginning of the prodromal stage after about 1 to 2 weeks. And as I already said, stepwise increase in fever. And as I said, prodromal stage means not yet really those very disease-specific symptoms, but rather general symptoms. And what we have here, again, fever rises, and then these rather unspecific symptoms, so general malaise, headache, abdominal discomfort. There are a couple more that you could mention here, but the takeaway for this is rather general symptoms, so you wouldn't at this point necessarily know, okay, that's typhoid fever. Then acute stage, and this is where the symptoms were, of course not all patients again, but where many patients also develop these much more indicative symptoms of that it is typhoid fever. And as I said, what we have here is this constant and often rather high fever. Then we have delirium. We have hepatosplenomegaly, so enlargement of the liver and of the spleen. And very characteristic of typhoid fever are these rose spots, these so-called roseolae, which are mainly found, or which are found on the abdomen. And again, rashes obviously occur in different diseases, but these ones are really very characteristic of typhoid fever. And then we can have, this is more towards the end of the acute stage, even reaching in the deferescence, convalescence stage, the possibility of an intestinal hemorrhage or even perforation of the gut. And what we also have with typhoid fever, again, rather towards the later stages, is this so-called pea soup diarrhea. So both in the Salmonella Enteritis there is diarrhea, and also with typhoid fever there can at least be diarrhea, but again with the Enteritis this is much earlier. Typhoid fever, it's rather late, and also the diarrhea itself is different between those diseases. So Enteritis, it doesn't always necessarily have to be bloody, but this is typically this watery to slimy to bloody diarrhea. And typhoid fever, it's this what's described of in terms of consistency and also color as this pea soup diarrhea. Okay, final stage is marked by a falling fever, but again, even still during this stage, even so the patient is recovering, there may be these longer term problems or later problems, and the most severe one, probably one of the most dangerous things about typhoid fever is this possibility of gut perforation. And then, in the best case, the patient fully recovers. In the worst case, the patient doesn't make it, and a certain number of patients, especially if untreated, remain carriers. So they still have the Salmonella, they keep excreting via their stool the Salmonella, but don't necessarily have symptoms anymore. And now, again, we had this disease with rather wide range of symptoms. In the prodromal stage, as I said, we could add even more symptoms here, more of these general symptoms. In the acute stage, many are very specific symptoms. And now we want to try to explain on a molecular cellular level, where do these symptoms come from? Okay. Let's now try to explain those varying symptoms on a cellular level. I've shown here on the left 3 of the stages. I have left out the convalescent stage, but the first 3 are shown, and we will go through them and try to explain what happens in which stage. We'll begin with the incubation period, and actually surprisingly much already happens in this period, even though we don't observe any symptoms here. So we start with our bacteria being in the gut lumen. And they enter through these cells here, which are the so-called M cells or microfold cells, which are cells dedicated to sampling from the lumen, and they pass those antigens or sometimes complete cells on to immune cells below. Those immune cells are dendritic cells or macrophages, and they reside in what is shown here is called the Peyer's patch. To some extent, bacteria can also enter through the normal mucosal epithelial cells, but this entry through the microfold cells is an important one. Okay, so the cells are now within those dendritic cells, macrophages. They are next going to be transported to the draining lymph nodes, which are the mesenteric lymph nodes. Now, lymph nodes represent the place where they can go from the lymphatic system into the blood. So still within those dendritic cells and macrophages, the Salmonella now get into the blood, and they will end up primarily in organs of the so-called MPS, so mononuclear phagocytic system, or RES. RES is the older name, but which is actually still used more frequently, the so-called reticuloendothelial system. Which organs are these? So which are the organs of the RES? That's the liver, the spleen, and the bone marrow. What do these organs have in common? They have in common that they have a vasculature which is rather irregular and rather fenestrated, which is often referred to as sinusoids. So if we take the liver here as an example, this functional unit that's shown here is the so-called lobule, which has this kind of hexagon structure, and we see here blood flow from the so-called portal triad to the central venule, and you see here this relatively irregular and fenestrated capillary. And what's also typical for the liver and spleen in particular is that you see those immune cells here, so these tissue macrophages, which in the liver are called Kupffer cells. And this is why the liver and the spleen are filtrating organs. So they filtrate the blood, take out what should not be in the blood. And even though our phagocytic cells that now carry the Salmonella, they don't really manage to efficiently present antigens because the Salmonella prevent the fusion of phagosome and lysosome, similar as the non-typhoidal Salmonella, as the Enteritis Salmonella are doing that. And they also generally prevent apoptosis of those cells, but nonetheless those cells can to a certain extent show that something is wrong with them. This is, for example, by not expressing or less expression of the so-called don't eat me signals, and by expression of eat me signals, something like phosphatidylserine. So they show to these Kupffer cells, to these tissue macrophages, that something is wrong with them, that they should be filtered out. Through this, the Salmonella again end up in phagosomes and again can prevent the fusion of the phagosome to the lysosome, so they're not getting destroyed. So they're now in the organs of this RES and multiply here within phagocytic cells, within macrophages. At some point, and this is now, here we are transitioning from the incubation period without symptoms to the prodromal stage. At some point, they will have reached a critical mass, the Salmonella will have reached a critical mass, and those macrophages will die, and the Salmonella will be released into the blood. At first, this is an intermittent release, so that means they're released into the blood, then they're taken up by again phagocytic cells. They get free and so on, so this can repeat a couple of times. So we can imagine, if we say this is the load of the bacteria in the blood, it may be something like this. Will also increase then over time because more and more cells are infected and the bacteria get more and more. And this is probably linked to this stepwise increase in fever. Again, the bacteria are not persistently in the blood, but it's intermittent. So, as we said, symptoms we see here is fever, and these general prodromal symptoms. And what I mean by prodromal symptoms is rather general symptoms of systemic inflammation, through those cytokines that signal systemic inflammation, like IL-1, IL-6, TNF-alpha, and main consequence fever, but also general malaise, headache, and so on. What happens next? So the Salmonella will keep proliferating, and eventually we find them in the blood, but now in a sustained manner, so they're never completely eliminated from the blood again. And therefore now we also have this not intermittent fever anymore, but this long-lasting fever, and this fever which can be very high, so which can be 39, 40 degrees. And this state of continuous systemic inflammation is then also what leads to the so-called typhoid state. Which reflects those neurological problems, and the main one here, delirium. Or the namesake one for typhoid fever, at least, because typhoid meaning fog, so the patients can't fully use their mental faculties anymore. They feel like they're in a fogged up state. Okay. Other kind of symptoms that we now observe, we have transitioned to the peak stage now. There is, on the one hand side, we have said those bacteria have been in the liver and in the spleen, and this here we will have more and more inflammation, and we will have more and more proliferation of immune cells, which can then be reflected in hepatosplenomegaly. A very typical symptom for typhoid fever are these so-called roseolae, the rose-colored spots. This comes from the bacteria that are in the blood. What happens here is, so the bacteria have been present now in this organism for several weeks. The immune response has been mounted, and at this point we even have the adaptive immune response, meaning we have antibodies. Those antibodies will lead to a clumping of those bacteria in the blood, and then these clumps can get stuck in capillaries and lead to further inflammation there. And if those capillaries are right beneath the skin, then this inflammation in the capillaries can be seen as these rose-colored spots. This symptom of this roseola in the abdominal area is very typical for typhoid fever. We don't typically see that with other bacteria, and this is linked to the differences in bacteremia with other bacteria. So for most other bacteria, once you have them in the blood, then they multiply quickly in the blood, and there is also a strong immune response to this bacteria, leading to these typical symptoms of sepsis. So this very strong inflammation associated with a decrease in blood pressure, and this is not typical for Salmonella. So for Salmonella, they enter the blood more gradually, as we have seen, and then we don't have this very sudden onset of this strong systemic inflammation. What else do we have? So we said the bacteria were initially already in the Peyer's patches, so they may have proliferated further here, but probably there is also a secondary infection of the Peyer's patches, and this is what leads, or general inflammation of the gut. This leads to this is also what's seen as a relatively typical symptom for typhoid fever, this pea soup diarrhea. Not completely typical just of typhoid fever, but at least sets it apart from the Salmonella-based enteritis, which is typically more this diarrhea, slimy, bloody diarrhea. So what's happening here? Inflammation in the gut. This leads to more permeability, water permeability of the gut, so loss of water through the epithelium, and on the other hand side also more gut peristalsis. The food is moved through the gut more quickly, and this is associated with this pea soup color. Another complication of this strong inflammation, that's again in contrast to the non-typhoidal Salmonella that occurs late here in typhoid fever, is this danger of gut perforation. I'm writing that very far down because this is something that can occur really late, that can occur even during the convalescent stage. So even while the patient is getting better, this gut perforation can still occur, and this is one of the most feared symptoms of typhoid fever. And finally, one more. We can also have an infection of the gallbladder now by these bacteria. And this is so important because this is a place where those bacteria reside long-term in these long-term carriers. And from there, of course, they can then enter the gut and be distributed from here and passed on to the next patient. I want to only very briefly also talk about treatment, just because this also differs between the Salmonella enteritis and the typhoid fever. So typhoid fever, to give the patient the best chance of recovering, as we said, you treat with antibiotics. These ones that are listed here are from the CDC, so there is probably some location and some probably also time dependency, so depending on resistances, those may change. So if you're interested in this topic, then yeah, just look up this link. And then importantly, for the Salmonella enteritis, just for the regular salmonellosis, the treatment recommendation is actually supportive therapy, but don't give antibiotics. And the reason for this is probably mainly because those Salmonella, in the gut or at the gut lumen, at the gut epithelium, they are not just fighting against the immune system, but they are also competing with other bacteria that are there. Antibiotics damage the microbiome, so they actually probably give these Salmonella a better chance. So antibiotics may lead to a prolonged shedding and potentially to a worse disease outcome. Importantly, there are exceptions. So, for example, children, elderly, immunocompromised. Okay. I want to come back briefly to Salmonella typhimurium, because we said this may be confusing that it's called typhimurium, even so it's in the non-typhoid group. Actually, it is the case, it doesn't cause typhoid fever in humans, but it causes at least a similar, illness similar to typhoid fever in mice, and that's why it's called murium. So murium, like murine, meaning in mice. The complete story is it doesn't cause this typhoid fever-like infection in all mice, but only in certain strains of mice, particularly in mice that are mutated in a certain way in their immune system, and in particular these are laboratory strains that are even deliberately mutated in this function. Nonetheless, my take is that for having this systemic Salmonella infection, these Salmonella need to be very well adapted to the host. And we see this, for example, from the typhoidal Salmonella, which we find only in humans. So those are not the ones that we find in the chickens on the chicken meat, on the chicken eggs, but we find them in only humans, and there is where they cause this systemic infection. So there seems to be a similar story with typhimurium. Even so, they cause infections in humans. It's not generally a systemic one, unless immunocompromised people and so on. But okay, maybe this is not the best example because of this. Okay, it's only in certain strains of mice. Better example may be Salmonella gallinarum, which is known for causing a typhoid fever-like illness in chickens, but in humans it doesn't really do much, or only causes milder infections in humans. We have looked at the different diseases. So on the one hand side, the Salmonella caused enteritis, and on the other hand side, the Salmonella caused typhoid fever. We can go back to our initial question. Why are the diseases caused by different Salmonella, so enteritis versus typhoid fever, so different? And so what we saw is the enteritis causing Salmonella, they cause this very strong local inflammation at the lamina propria, so still very close to where they enter the gut. And that's responsible for the symptoms that we see. We said, okay, in rare cases, in immunocompromised cases, this Salmonella can spread systemically, but typically they do not. Typically they cause this local inflammation. Versus the typhoid Salmonella, it's often said they take a stealth approach, so they get through the intestinal mucosa relatively unnoticed, and then they spread throughout the system. The disease is a systemic disease. What are virulence factors that may be causing this difference? And so in the very beginning, in the motivating question, we talked about these surface factors, and we said, okay, O and H antigen and the capsule. And in Salmonella, the capsule antigen is called the VI capsule, for virulence capsule. And very interestingly, this is only found in the typhoid strains. So it seems likely that these different modes of action, modes of pathogenicity have something to do with this VI capsule. That seems to be an important factor. There are some counter arguments against this, because even though the VI capsule is found in typhoid strains, it's not found in all the typhoid strains, or let's say at least in the paratyphoid strains, paratyphoid, which is very similar to typhoid. There the VI capsule is actually not found, and it was also tried experimentally, so typhoid strains where the VI capsule was deleted, they still caused an illness which resembled typhoid fever more than enteritis. Interestingly, this was an experiment that was done in human volunteers indeed. So the reference that I'm giving down here is not the study itself, but it's a review that's talking about this difference between enteritis and typhoid Salmonella diseases. So it seems to be not only this VI capsule, even though the VI capsule plays an important role. And something else that seems to be there, it's not just the typhoid Salmonella, which are very stealthy, but somehow that these Enteritis Salmonella are deliberately, let's say, humanizing it a bit, deliberately causing this inflammation. So they seem to want to recruit those immune cells. They want to cause this inflammation, and a theory is that this then leads to the killing of other bacteria in the gut, whereas the Salmonella can defend themselves better against this inflammation, and so they get all these they get this niche more or less for themselves, because the other bacteria are killed and they can survive pretty well in this niche. Nonetheless, it seems like this VI capsule is an important virulence factor and an important one for distinguishing the typhoid versus non-typhoid Salmonella. So if you've been paying attention, then probably this question here comes to mind, which is our second big question for today. How does it make sense that the main virulence factor of a bacterium, so Salmonella typhi, that is spread via phagocytes is anti-phagocytotic? So this virulence factor is, of course, the capsule. How does it make sense? We said these phagocytes, these macrophages, they are so important for the pathogenicity of Salmonella typhi. How does it make sense that this bacterium has a virulence factor that actually, where the main job is defending against phagocytosis? And that's actually something that bothered me for a long time, and I've been teaching this course and it has been bothering me since, and fortunately nobody ever asked me this question, but now I think so most of what I told you so far is what you can find in the textbooks. So this is hopefully something where I can tell you something new and an explanation that hopefully makes sense for this question. Okay, so the answer can be found in this paper that I'm citing here. It's maybe not the complete, not the only answer, but it's a very impressive answer for this riddle. So we're going to start off with the non-typhoidal Salmonella, the ones that don't have the capsule. And as this paper describes, a typical immune response in a person that has not seen these pathogens before will be via IgM. So IgM, in this case, these are antibodies that here are not produced via T helper dependent response, but via a T helper independent B cell response. So they recognize repeating patterns on pathogens. They will then be detected, so they will bind the bacteria and will be detected by complement proteins, and therefore lead to opsonization of this bacteria. In addition, there are also these fragments of the complement proteins that then serve to attract as chemokines further immune cells, and in particularly neutrophils. So since these bacteria now have been opsonized, they will be taken up by phagocytes, so both here by this macrophage and by this neutrophil here. I should say this is probably not the only way via the complement receptor, but so for example, these bacteria have flagella, they have LPS, so also just regular uptake via toll-like receptors will play a role here because like the LPS is not masked by a capsule. What should still be said is that these neutrophils then they are known for producing radical oxygen species and therefore leading to this tissue damage that's very much associated with this local inflammation in the Salmonella caused enteritis. Now turn to the typhoidal Salmonella. So those are the ones that have this capsule. Capsule, main role of a capsule is being anti-phagocytic. On the other hand side, these bacteria rely on being phagocytosed by macrophages, by dendritic cells for their pathogenicity mechanism. So how does this make sense? So we said they have the capsule, so IgM is not going to bind them. They are not going to be taken up via the toll-like receptors on the macrophages and on the neutrophils because of the capsule. Now comes the interesting point. The macrophages express a receptor called DC sign, which is not a toll-like receptor, but is actually a lectin. And via this receptor, they're able to bind the typhoidal Salmonella and to take them up. Now, since this is a lectin and not a toll-like receptor, this is not a receptor that's associated with a signaling mechanism that would lead to inflammation. Also interestingly, this receptor is found on macrophages, but apparently not on the neutrophils. So the typhoidal Salmonella will not lead to this ROS production and to this tissue destruction that the non-typhoidal Salmonella are associated with. Even more interestingly, it's not just that this uptake in the macrophages doesn't trigger an inflammatory response, but it seems to even trigger an anti-inflammatory response. Because you might say, okay, well, these bacteria still have the flagella, this still might lead to inflammatory response via TLR5, but actually when at the same time the capsule polysaccharide is detected via this DC sign, there seems to be even an anti-inflammatory response triggered, leading to IL-10 production. And so this seems to a large part to be the answer to our question. With this, let's summarize what we learned in this course today. So we talked about Salmonella and we said, even though they're all, well, mainly one species, there are different reservoirs and these reservoirs can behave very differently. On the one hand side, we have the non-typhoidal reservoirs, the non-typhoidal Salmonella, and they cause a illness that's characterized by enteritis and diarrhea, and this comes from local inflammation in the gut. And then we have those typhoidal reservoirs, those typhoidal Salmonella, where the disease looks very different. So this is a systemic infection, and this is associated with the survival of this Salmonella in macrophages, also in dendritic cells. Somewhere early on I may have said in phagocytes. That's too imprecise because we then talked about this very important point about the role of this VI capsule, which prevents the phagocytosis via neutrophils. So these phagocytes, there indeed the phagocytosis is prevented by the capsule. And it also prevents the inflammatory response. So this is kind of a function as you would expect from a capsule, that it prevents phagocytosis. However, it triggers then phagocytosis by macrophages and also by dendritic cells. However, in this case, via a non and key references that we used here. So the first one is this very interesting paper that talks about the non-inflammatory phagocytosis of typhoid Salmonella by macrophages via this DC sign, so via this lectin. The second one is a review comparing non-typhoidal to typhoidal Salmonella. And then the third one is kind of a further reading. So I mentioned briefly that the non-typhoidal Salmonella in a way deliberately cause inflammation, this local inflammation. And this paper shows an example how these non-typhoidal Salmonella benefit from this inflamed environment. Last but not least, the image credits. So shout out to all these generous people that are providing their images for free via Wikimedia Commons.